Today we're going to talk about DNA. But what is DNA? DNA is made from deoxyribose nucleic acid. Nucleic acid is made out of nucleotides whose specific function is to carry genetic information. This DNA is in a double helix shape. So now that we know the basic idea of DNA, we can look deeper into its structure. We have two main parts in DNA, the backbone and the bonds. We are first going to talk about the backbone. It's made from a phosphate bond and a deoxyribose pentose sugar. Deoxyribose pentose sugar is shaped like a little house. Now for the bonds. There are 4 nitrogen bases in a DNA molecule: guanine, cytosine, adenine, and thymine, or GCAT. GCAT will tell you the initials of the bonds and the complementary bases. G goes with C and A goes with T. Why do these go together? Well, because these go under the classifications of purines and pyrimidines. A good way to remember is only the pyrimidines have the letter Y in them. One pyrimidine and one purine go together. However, these pyrimidines and purines are like enzymes, or puzzle pieces. They need a specific piece in order to fit a bond. There are 2 types of bonds: hydrogen bonds and covalent bonds. Covalent bonds are strong bonds that hold the deoxyribose pentose sugar and the phosphate bond together. Hydrogen bonds happen when nitrogen base pairs adenine to thymine and guanine to cytosine. Hydrogen bonds are weaker, so we may replicate DNA. There are 4 simple steps to replicate DNA. A helicase enzyme comes and unzips the genes. See what I did there? Nucleotides are little floaty nitrogen bases, and they begin to find their nitrogen base complement. A polymerase connects the paired nucleotides to the original strands of DNA. After ligase fills the gaps, 2 molecules of DNA are created. However, this process is a little funny. When DNA splits, the new copy is being filled in backwards. The first strand, the leading strand, goes from 5' to 3'. This means that the DNA is being unzipped. A new copy is being filled from the bottom up. The second strand, the lagging strand, moves from 3' to 5'. This means that the DNA is being unzipped and a new copy is being filled from the zipper down. This strand is moving backwards. And that is the basic breakdown of DNA and the DNA replication process.